Good morning students. Welcome to the world of SciTech, Science and Technology. Today we are discussing about RNA. In the previous class we have already discussed about DNA. Because of this current situation, this pandemic situation, I also faced some difficulties. That's why I had to be very late to take this class. Sorry. So let's move on to our topic, RNA. RNA is ribonucleic acid. It is similar to DNA, but it is a single-stranded molecule. RNA's backbone is made up of a sugar group called ribose and a phosphate group. And the base sequences are adenine, guanine, cytosine and uracil instead of thymine. Different type of RNA exist in the cell. They are mRNA, tRNA and rRNA. These three type of RNA are very important in protein synthesis. What is the primary function of RNA? RNA is to convert genetic information stored in the DNA into proteins to carry out cellular functions. That is, transfers information stored in the genome into proteins. And the process is called translation. We will discuss about later. DNA provides the code for the activities. We know that base sequences code the genetic information in the DNA. But RNA converts this code into proteins. That is the difference between DNA and RNA. We heard about DNA replication, transcription and translation. What it is? From existing DNA to make new DNA is called DNA replication. From DNA to make RNA is called transcription. From RNA to make new proteins is called a translation. Proteins are the building block of life. We will discuss about later. RNA is synthesized from DNA by the help of an enzyme RNA polymerase. Term end with the ASE. It is an enzyme. Here it is RNAP. What is the relationship between RNA and ribosome? We know that RNA is a nucleic acid and it is a macromolecule, while ribosome is a cytoplasmic organelle. RNA carries the genetic information that is translated by ribosome into proteins to carry out cellular functions. It is the relationship between RNA and ribosome. Ribosome is composed of approximately 60% of rRNA and 40% of ribosomal proteins. That is, rRNA is associated with proteins to form ribosome. It is the function of rRNA. tRNA carries specific amino acid to the ribosome to form polypeptide chain. mRNA carries a coding instruction of amino acid of a proteins. This is the function of mRNA, tRNA and rRNA. Students, we move on to transcription. Okay students, let's move on to that topic. Students, the central dogma of molecular biology is the information available in the DNA is passed to messenger RNA, which is then used for the synthesis of a particular protein. We know that each DNA molecule is made up of two strands and there are 4 nucleotides present in DNA: adenine, cytosine, thymine and guanine. And each of the nucleotides on one side of the strand pair with a specific nucleotide on the other side of the strand. And this makes up the double helix with the help of sugar and phosphate backbone. Adenine always bind to thymine, while cytosine and guanine always bind to one another. This relationship is called a complementary base pairing. We always heard about this word in molecular biology, the complementary base pairing. Nucleic acids, that is DNA and RNA, are made by nucleotides, but proteins are made by 20 small molecules of amino acids. When the transcription process start, the process start from initiation process. We can hear about promoters. What are the promoters? There are certain specific areas on the DNA that act as starting signals for initiation process. The RNA polymerase, that is the enzyme helping for transcription, attaches at the promoter site on the template DNA strand. In human beings, about 10 raised to 5 transcription sites are present on the entire DNA. Factors required for promoter clearance are every double-stranded template bearing a TATA box, TATA box, and some binding proteins TBP, TF2B, TF2F, TF2E, and TE2H. What is promoter clearance? RNA polymerase must clear the promoter once the first bond has been synthesized. That is the promoter clearance. Okay. We know that transcription means from existing DNA to make new RNA to produce proteins. Okay. So that template strand means the strand that is used during transcription to produce RNA. One strand of the DNA is used for the transcription. That strand is called a template strand. What is coding strand and non-coding strand? Only one strand is used as template for transcription at any given time. This template strand is called a non-coding strand. The non-template strand, the other strand, is referred to as the coding strand because its sequence will be same as that of the new RNA molecule. We know that DNA is referred to as the genetic code for life because it contains information about which amino acids join together to create different proteins. In molecular biology, we can heard about a codon. What is codon? A sequence of 3 nucleotides which together form a unit of genetic code in a DNA or RNA molecules. Codon is a consecutive sequence of 3 bases. One codon is MET and the other one is TRP. In mRNA, for example, UUU codes for phenylalanine. What is proteins? Any class of nitrogenous organic compounds which have large molecules composed of one or more long chains of amino acids and are essential part of all living organisms, especially as structural components of body tissues such as muscles, hair, etc., and enzymes and antibodies. So that is a codon and proteins. Transcription taking place all the time, only certain areas of the DNA are copied. That is, selected regions on the sense strand are copied. This is taking place Xerox copy of particular page of the book like that. So the genetic information of DNA is transcribed or copied to the mRNA. During transcription, the message from the DNA is copied in the language of nucleotides, that is 4-letter language. So during transcription, a message from the DNA is copied to the mRNA. RNA polymerase uses one of the DNA strands as a template to make a new complementary RNA molecule. Transcription ends in a process called a termination. Transcription can be broken into 5 stages: pre-initiation, initiation, promoter clearance, elongation, and termination. The first step is pre-initiation. In this step, the RNA polymerase, that is the enzyme helping for the synthesis of RNA, and cofactors bind to DNA and unwind the strands of DNA, creating an initiation bubble. This space grants RNA polymerase access to a single strand. That means an initiation bubble will help the RNA polymerase to enter the single strand of the DNA molecule. Approximately 14 base pairs are exposed at a time of the pre-initiation stage. What is mRNA? mRNA is a single-stranded RNA molecule that is complementary to one of the DNA strands of a gene that leaves the cell nucleus and move to the cytoplasm where proteins mold. So that the first RNA formed in the transcription process is mRNA. Initiation stage of transcription. In this stage, the DNA helix partially unwinds and the RNA polymerase enzyme binds the promoter site on DNA with the help of sigma factor and moves forward in the template strand. When it reaches the appropriate site on the gene, the first nucleotide of the mRNA attaches to the initiation site of RNAP. This becomes the 5' end of the mRNA. It will be complementary to the base present in the DNA at that site. The sigma factor is now released. The next nucleotide attaches to the RNAP. A phosphodiester bond is formed. Then the enzyme moves to the next base on the template DNA. In mRNA, three nucleotide units called codon dictates a particular amino acid. For example, AUG code for the amino acid methionine. So this is done in initiation stage of transcription. In the elongation stage of transcription, the RNA polymerase enzyme moves along the DNA template. New nucleotides are incorporated in the nascent mRNA one by one according to the base pairing rule. That is, adenine in DNA is transcribed to uracil in mRNA, and guanine to cytosine, cytosine to guanine. The synthesis of mRNA is from 5' to 3' end. That means the reading of template DNA is from 3' to 5' end. So the elongation process is going on. That time the mRNA molecules, to the mRNA molecules, new nucleotides are added from DNA template. The last stage of transcription is termination. The term means everything. That means the specific signals are recognized by a termination protein. The RHO factor, that is the prokaryotic protein involved in the termination of transcription. RHO factor catalyzes the dissociation of mRNA from genomic DNA and the RNA polymerase. When it attaches to the DNA, the RNA polymerase cannot move further. So the enzyme dissociates from DNA and consequently newly formed mRNA is released. So in the termination process result in the release of newly synthesized mRNA from the elongation complex. So in transcription process, the information in a strand of DNA is copied into new molecule of mRNA. This newly formed mRNA copies of the gene then serves as blueprint for protein synthesis during the process of translation. During translation, reads the genetic code in mRNA and makes a protein. Children, this is the transcription process. I think all of you understood very clearly. Students, in the next class we will discuss about translation. Okay students, God bless you.